Hi everyone, in this video we are going to talk about cellular respiration, and we are specifically going to talk about the second and the third step, which are called the PrEP step or the preparatory reaction, and the citric acid cycle or the Krebs cycle. So these two reactions will occur only if oxygen is present within the cell, and they occur after glycolysis has already taken place. So you'll remember that glycolysis takes place in the cytoplasm. It does not require oxygen or any specialized organelles, and it produces 2 molecules of pyruvate. And as a reminder, those 2 molecules of pyruvate each have 3 carbons in them. Okay, and so if oxygen is present, pyruvate is going to enter the matrix of the mitochondria, and this is going to be converted into something called acetyl-CoA. Now, acetyl-CoA is a 2-carbon molecule that is attached to a CoA enzyme, and this is what we are going to call the PrEP step. Okay. So from the PrEP reaction, you'll notice that we actually have to go in here and say that we have 2 molecules of acetyl-CoA being produced. And the reason why we have to stick a 2 in here is because we had 2 molecules of pyruvate that are going to be entering the PrEP step. So for every molecule of glucose, the PrEP step and the Krebs cycle or the citric acid cycle are going to occur 2 times. You'll notice that we lose carbons because we go from a 3-carbon molecule to a 2-carbon molecule, and we are going to show that as 2 molecules of carbon dioxide are lost. So if you think back to the cellular respiration equation, our byproducts are going to be water and carbon dioxide. In photosynthesis, remember that carbon dioxide and water were used to make glucose. Therefore, in order to break down glucose, we have to release carbon dioxide, and then we'll create water as a byproduct. Now, carbon dioxide is completely lost during the PrEP step and during the Krebs cycle, and your water is going to be created in the electron transport chain that I'll talk about in the next video. So the process of converting pyruvate into acetyl-CoA is oxidation, therefore something has to be reduced. So we're going to bring in 2 molecules of NAD+, and those are going to be reduced to 2 molecules of NADH. As a reminder, NADH is an electron carrier, so it's carrying the electrons, and then those electrons are going to be energized, and then that energy is going to be used during the electron transport chain. And again, we'll talk about that in the next video. So now we have entered the Krebs cycle. Okay, remember that is also known as the citric acid cycle. And because it is a cycle, something is recycled. Just like in the Calvin cycle of the dark reactions in photosynthesis, RUBP was recycled there. It's a 5-carbon, and it picked up a 1-carbon carbon dioxide. Here we have 2 carbons entering, and so we have a molecule called OAA, and this is just a 4-carbon molecule here. Okay, so OAA is going to pick up acetyl-CoA, and we are going to have citrate being produced. And citrate is a 6-carbon molecule. All right, so the next step of the process, we're going to take citrate and we are going to convert it into intermediate 1. The name is not super important for any of these intermediates. There's going to be 4 of them. But notice we have 2 molecules of citrate being produced because we have 2 acetyl-CoAs, which means we're also using 2 OAAs, and we're producing 2 intermediate 1s. Intermediate 1 is a 5-carbon molecule. So you'll notice we're going from a 6-carbon molecule to a 5-carbon molecule. So we are having 2 molecules of CO2 being given off here, 1 for each of the molecules of citrate. So as we have oxidation occurring going from citrate to I1, we have reduction, and that is going to be in the form of NAD plus to, 2 molecules of NAD plus to 2 molecules of NADH. Okay, and then we are going to take our intermediate 1 and convert it into intermediate 2, so we get 2 molecules of intermediate 2. Again, we have lost carbons because this is a 4-carbon molecule, so we have to lose 2 more molecules of CO2. And then as this is oxidized, we are going to reduce 2 more molecules of NAD plus to create 2 more molecules of NADH. Alright, so you'll notice at this point we have actually lost all 6 carbons from our original glucose. We've lost 2 up here during the prep step, then we lost 2 more converting citrate into intermediate 1, and then the last 2 converting intermediate 1 into intermediate 2. So you'll see that we have a 4-carbon molecule for intermediate 2 as well as a 4-carbon molecule for OAA. So from here on out, intermediates 3 and 4 are simply going to be reconfigured so that we can get back from intermediate 2 to OAA. And in the process, we are going to have some more oxidation reduction reactions occurring. And while I'm only going to show you intermediates 3 and 4, there are multiple steps that we're going to skip in this, simply because these last ones that I'm going to show you, the last 2, are the ones that are creating products that are important for the electron transport chain and for creating energy. The others don't do that. So these are the most important ones. We're going to go from intermediate 2 to 2 molecules of intermediate 3. And at this step, we're actually going to make ATP. So you have 2 molecules of ADP plus P, and they are going to come in and be kicked out as 2 molecules of ATP. In the very last, in the next step, we're going to go to 2 molecules of intermediate 4. And here we're going to create a different molecule that we have not talked about yet. We have 2 molecules of something called FAD, and they are going to be kicked out as 2 molecules of FADH2. So FADH2 is similar to NADH. It is an electron carrier that is going to be used dropping off electrons and exciting electrons in the electron transport chain. Then for the very last step, we're going to take our intermediate 4 and convert it back to OAA. Okay, and I didn't quite leave myself enough room here, so I am going to erase this up here and I'll draw it right back in, but up a little higher. Okay. So I'll draw our 2 molecules of NADH up here. Alright, and then when we are going from intermediate 4 to OAA, we have 2 more molecules of NAD plus being converted into 2 more molecules of NADH. Okay, so the whole process of the Krebs cycle is to get rid of our carbon dioxides as well as to produce our electron carriers that are going to be used in the electron transport chain. So the electron transport chain is going to be the most efficient, highest producing reaction in cellular respiration as far as production of ATP goes, and that is because of all of these high energy electron carriers that we've got produced from the Krebs cycle. So I'm going to go through here and I'll use a blue color to show what our yields are. So first let's look at our carbon dioxide. As I said, we got 2 molecules of carbon dioxide in the prep reaction, then we created 2 molecules of carbon dioxide going from citrate to intermediate 1, and then 2 more molecules of carbon dioxide going from intermediate 1 to intermediate 2. So we have given off our 6 molecules of carbon dioxide that are mentioned in the cellular respiration equation. The next molecule that we're going to look at is our NADH. So we have 2, 4, 6, 8 total NADH being produced between the PrEP and the Krebs. So the Krebs cycle produces 6 molecules of NADH. The PrEP step produces 2 molecules of NADH. So you get a total of 6 NADH. You'll notice too that we also get 2 molecules of FADH2, and that is produced during the Krebs cycle. So we have 2 FADH2. So these 6 NADH, as well as the 4 NADH that were produced from, oops, I'm sorry, this should have been an 8. Huh. Man, don't you love math? Okay, so this here needs to be an 8. So we have 8 total NADH being produced from the Krebs and the PrEP step. Along with the 2 NADH from glycolysis, we're going to have 10 NADH that are going to enter into the electron transport chain. Then we also have these 2 FADH2s. So between the 12 molecules, we're going to end up dumping a lot of electrons into the electron transport chain. And then finally, when we're talking about yields, it's important to note that we have these 2 ATP being produced here during the Krebs cycle. Remember, we also had a net yield of 2 ATP being produced from the from glycolysis. All right, so in the next video, we will talk about the electron transport chain.